So, to discuss the electron transport chain and oxidative phosphorylation as a whole, I want to consider the proteins in more detail and start our conversation with a general overview of what the entire process looks like, as well as discuss some important principles that govern these proteins. This will allow us to introduce some terminology and make the details of each different proteins easier to digest. Now, the way these proteins are organized is convenient for a metabolic map, but not so much for this section. As such, let's reorganize all the proteins in a manner that will be insightful for us. For that, let's take a moment to understand where we are. The top beige color represents the cytosol, and the bottom part is the mitochondrial matrix. The cytosol and the mitochondrial matrix are separated by two mitochondrial membranes, an inner and outer membrane, and the two create the intermembrane space. The proteins that we are concerned with are all embedded in the inner mitochondrial membrane, and as you can see, now they are arranged in a linear order. This entire machinery that has pretty much the sole purpose of generating energy for the body can be broadly divided into two functional roles. The four first proteins on the left, which are typically called complex 1, 2, 3, and 4, mediate the electron transport chain. The last remaining protein, ATP synthase, and various transporters mediate oxidative phosphorylation. The electron transport chain and oxidative phosphorylation phases sequentially work together to make ATP, and everything begins with our NADH and FADH2 molecules that we have made during glycolysis, the pyruvate dehydrogenase complex, and the citric acid cycle. Also, as a quick reminder, since it's important to keep this terminology in mind for this section, oxidations are a loss of electrons and reductions are a gain of electrons. So, if we first consider NADH, the molecule gets oxidized by complex 1, which produces back NAD+. Given that this is a redox reaction, the oxidation of NADH to NAD+ is coupled to the reduction of complex 1. Through a very intricate mechanism that we will cover shortly, the electrons that are grabbed by complex 1 from NADH are passed on to a molecule called coenzyme Q. This passing of electrons generates a sufficient amount of energy for complex 1 to pair that process to the pumping of protons into the intermembrane space. Now, in a similar fashion for complex 2, the complex oxidizes FADH2 to FAD and transfers the electrons of this oxidation to coenzyme Q. The electrons from coenzyme Q are then passed on to cytochrome C to complex 3, which pairs this transfer to the pumping of protons in the intermembrane space. Finally, these electrons end up reacting with oxygen to produce water in complex 4, which here again generates enough energy to pump protons in the intermembrane space. If we briefly summarize what we just covered with the electron transport chain, this section has three main functions: oxidize NADH and FADH2, pump protons in the intermembrane space, and make water out of oxygen and received electrons. Also, to quickly define what coenzyme Q and cytochrome C do, the two molecules are responsible for carrying the electrons from NADH and FADH2 to complex 4. So, as you can see, the electron transport chain holds pretty well its name, as a big part of it is carrying the electrons to make water. Importantly, this movement of electrons generates the energy to pump protons in the membrane, and this will be a crucial element for oxidative phosphorylation. In this phase of energy production, the protein ATP synthase uses the proton gradient built by the electron transport chain to synthesize ATP out of ADP and Pi. We will obviously cover this mechanism in more detail shortly, but basically the energy that protons release when they cross the membrane back make ATP synthase rotate and force the synthesis of ATP. The last important proteins in this entire process are the transporters that import ADP and Pi into the mitochondrial matrix and export the ATP away so it can fuel the reactions that happen in the cytosol. So, to reiterate what oxidative phosphorylation is, it's the direct synthesis of ATP by the phosphorylation of ADP, which is fueled by the proton gradient generated by the electron transport chain. Now, before we discuss how each complex and protein mediates its function, I want to briefly touch on a term you are most likely to see in your classes, which is reduction potential. To define this term, reduction potential can simply be understood as the tendency of a substrate to want electrons. This reduction potential is a metric that can be quantified numerically, and the higher the value a substrate has, the more it wants electrons. For the purposes of this video, we are not going to consider the reduction potential values of each complexes, as it's not too important for us, but the main idea with the electron transport chain is that from complex 1 to 4, all the components are organized from the lowest to the highest reduction potential. This means that when complex 1 and 2 take the electrons from NADH and FADH2 and pass them on to the rest of the chain, the reduction potential for each successive step increases progressively until it reaches oxygen, which has the highest reduction potential out of all the elements in this chain. Because of this organization, it creates a few important implications. First, it makes the entire chain of events exergonic, or in other words, spontaneous. Recall that exergonic was a term we have covered earlier in our discussion when discussing the thermodynamic principles of metabolism. Here again, this term applies, and when an electron flows from a substrate with low to high reduction potential, the process is said to be exergonic. So, in the same way that we want glycolysis, the citric acid cycle, and virtually any other metabolic pathway to be exergonic, the electron transport chain also has to be exergonic such that it makes energy spontaneously. The way that the cell managed to accomplish this is to organize everything in this increasing reduction potential gradient. Another consequence of this reduction potential gradient is that it gives directionality to where the electrons are going. Since electrons naturally flow from low to high reduction potential, you can imagine that messing up with the order of the electron transport chain would disrupt the entire process of funneling the electrons to oxygen. As such, any toxin or mutation that inhibits the proper flow of electrons will have severe consequences on energy production and likely on the survival of the organism. Lastly, as you can see in the definition of exergonic, a reaction that is said to be exergonic will release energy along the way, so with the electron transport chain being an exergonic process, this is how it can continuously push protons into intermembrane space and create a gradient that is so important for oxidative phosphorylation. Alright, now that we get the gist of this entire system, let's consider each individual protein to understand how everything comes about. Let's begin with complex 1, also known as the NADH coenzyme Q oxidoreductase. As mentioned previously, the three general actions of this complex is to oxidize NADH to NAD+, reduce coenzyme Q, and pump protons in the intermembrane space. When we consider the structure of complex 1, it turns out that this very big protein carries out the redox reactions and proton pump functions at distinct sites. The two general areas of this protein are referred to as the peripheral arm, which is the segment that dips into the matrix, and the transmembrane arm. As noted here, the peripheral arm mediates the redox reactions, and the transmembrane arm mediates the proton pumping. If we first consider how the peripheral arm operates, the important idea here is that when NADH gets oxidized to NAD+ and gives away its 2 electrons to complex 1, the electrons reach coenzyme Q by being sequentially transferred from prosthetic groups. Complex 1 has two main types of prosthetic groups: flavin mononucleotide, which is simply abbreviated to FMN, and iron-sulfur clusters. Each complex 1 has one FMN, and mammals have about 8 iron-sulfur clusters. In terms of how they function, both types of prosthetic groups have a chemical structure that allows to accept and donate electrons. If we consider the structure of FMN, this molecule can interchangeably be oxidized and reduced, which allows it to pass electrons along. As such, the peripheral arm is essentially just a chain of redox reactions, and as we've just seen, all these prosthetic groups are organized from lowest to highest reduction potential to ensure the electrons get passed along to coenzyme Q. When coenzyme Q receives the 2 electrons, it becomes CoQH2. For the sake of space, I will not cover the structure of coenzyme Q just yet, but we will see it when discussing complex 2. Now, in my opinion, the interesting part about complex 1 is what happens in the transmembrane arm. It turns out that the way the protons cross the membrane to reach the intermembrane space is through channels that are incorporated in the structure of complex 1. Complex 1 has 4 of these channels, and the key that allows the protons to cross is the presence of polar amino acids such as lysine. If we consider the structure of lysine, a very important property of an amino acid of this type is that it can readily interconvert between its protonated and unprotonated form. As you can see here, lysine can either keep or let go of the hydrogen it has on the oxygen. More commonly, compounds that are able to do this are referred to as acids. Typically, the more a given compound lets go of its hydrogen, the more of an acid it is. One way to quantify this aspect is to consider the pKa of that acid, and the lower it is, the stronger it is. Again, for our purposes, we don't need to consider the exact values, but just get an intuition on how this process works. So, the idea here is that if you change the pKa value of a given substrate, it will change its affinity to what hydrogen it holds on the oxygen. This idea is exactly what is believed complex 1 does to pump protons across the membrane. So, to understand this process, let's start from the beginning even before NADH gets oxidized. In this state, the pKa of the lysine residues inside the first half of the channel make it so that lysines get protonated, or in other words, accept the protons. When NADH gets oxidized to NAD+, and eventually reduces CoQ to CoQH2, this entire sequence of events causes a conformational change in complex 1 that changes the pKa values and thus makes the lysine residues drop their proton. This proton gets accepted by lysine residues that are in the second half of the channel. When the complex returns to its oxidized state, the conformation of the protein changes again and the protons get dropped in the intermembrane space. Because there are 4 channels, the 2 electrons transferred by NADH to CoQH2 allow to pump 4 protons through complex 1. So, to put it again, the transmembrane arm couples the oxidation state of the protein to the pKa value of lysine and other polar residues, which causes this chain of reaction of proton pumping. As a last quick note on this process, the scientists that came up with this model of proton pumping have inferred the mechanism from crystal reconstructions of this protein. As shown by this reconstruction, complex 1 is pretty complex, and one element I'd like to point out, which I haven't in my rendition, is the transverse helix arm that you can see here as the purple ribbon. When I was mentioning that the transmembrane arm couples the oxidation state to the pKa, it is believed that it is accomplished via the mechanical movement of this helix. Obviously, if you want more information on this entire process, I would highly suggest consulting this article. They do a really good job at explaining their findings and model. Now, moving on to complex 2, the way I showed how this complex operates is a little misleading because, in contrast to NADH, FADH2 is not soluble. Instead, FADH2, which in its oxidized state is simply FAD, is located in complex 2. Recall that in the entire process, FADH2 was obtained from the oxidation of succinate to fumarate in one of the reactions from the citric acid cycle. In this reaction, this bond between the 2 carbons in succinate was converted to a double bond by the enzyme succinate dehydrogenase and released 2 electrons in the process that reduced FAD to FADH2. As you can see, succinate dehydrogenase is also part of complex 2. When it comes to what FADH2 does with the electrons afterwards, in a similar fashion to what we've seen with complex 1, complex 2 has a bunch of prosthetic groups including FAD itself and 3 iron-sulfur clusters. Here again, the electrons will flow from low to high reduction potential to reach coenzyme Q and reduce it to CoQH2. If we take a closer look at the structure of coenzyme Q, you can see that it has 2 main functional units. First, it has this ring which participates in the redox reactions and can be under 3 states: oxidized, radical, and reduced. In the radical state, CoQH has 1 electron, and in the reduced state, it has 2 electrons. The fact that coenzyme Q can hold 0, 1, or 2 electrons in a stable manner is an important aspect that will be relevant for our discussion on complex 3. Besides this redox ring, you can see that coenzyme Q has this long tail of hydrocarbons. In CoQH and CoQH2, this tail is omitted and simply replaced with R. Now, this tail that CoQ has is entirely made of carbons and hydrogens, which makes it hydrophobic. As such, this tail allows CoQ to be freely soluble in the membrane, and that is super important for the entire electron transport chain to work properly, given that coenzyme Q needs to move the electrons from complex 1 and 2 to complex 3. While we are on the topic of these structures, I want to quickly mention that FAD structure is basically the same as the FMN molecule we have just seen, but it has an extra AMP molecule attached to it. Regardless, the reduction and oxidation of the 2 molecules happen at the same place. If you recall our discussion on NADH shuttles, you might remember that the glycerophosphate shuttle used flavoprotein dehydrogenase to convert FAD to FADH2. This protein is also part of the electron transport chain to some extent, and as you might expect, the FADH2 that is produced within this protein serves to reduce CoQ to CoQH2. Finally, I want to mention that besides using FADH2 as an electron carrier, complex 2 differs from complex 1 as it does not pump any protons while it reduces CoQH2. This is why if we compare the glycerophosphate shuttle mechanism to the more traditional malate aspartate shuttle, you will see that the malate aspartate shuttle has the benefit of making NADH cross directly, which allows to pump some protons through complex 1. Nonetheless, even though no protons are pumped by FADH2 directly, the fact that it produces some CoQH2 is super important for complexes 3 and 4, which do pump protons. Alright, moving on to complex 3, also known as coenzyme Q cytochrome C oxidoreductase. This is where the electrons that we have accumulated in the form of CoQH2 will be transferred to cytochrome C. The process that makes this happen is called the Q cycle and allows 4 protons to be pumped in the intermembrane space. Before we get into the steps of the Q cycle, we first need to cover the structures of complex 3 and the molecules that will be important for this process. So, for the Q cycle to run properly, we will need 2 CoQH2 molecules, 1 CoQ molecule, and 2 empty cytochrome C molecules. With respect to complex 3, there are 3 important sites where most of the Q cycle will take place: the QO and QI sites, as well as the region where complex 3 interacts with cytochrome C. At the beginning of this cycle, the QO site will house CoQH2 and the QI site will house CoQ. In between all these groups, there are prosthetic groups that will be important for transporting electrons. In this complex, there is 1 iron-sulfur group and 3 variants of cytochrome molecules, cytochrome C1, Bl, and Bh. As you can see, there are different types of cytochrome proteins, but what differentiates them is not super relevant for our discussion. What is important for us is what makes them cytochrome proteins in the first place, and that is the fact that these proteins carry a heme group. If we consider the structure of cytochrome C, a heme group has 2 parts: a porphyrin group, which you can see in green, and importantly an iron ion in the middle that is shown in orange. The iron ion is what allows this entire molecule to be oxidized or reduced. In the oxidized state, the iron ion has a +3 charge, but it can receive an electron and become a +2 charge. An important aspect in this entire molecule is that the porphyrin group contains a lot of double bonds, which implies that the electrons across the structure of cytochrome C are highly delocalized. In chemistry, delocalization of electrons is also known as resonance, and it simply means that the electrons are not anchored to an atom and can move loosely around the bonds that are shared. For a prosthetic group such as cytochrome C, this is very good because it helps the electron transfer reactions occur. Although I pointed out this aspect here, if you go back and take a look at the structures of NADH, FMN, FADH2, and coenzyme Q, they all have rings with shared electrons, which facilitates all these electron transfer reactions. Now, in comparison to CoQ, cytochrome C can only hold 1 electron at a time, and this will be important to take into account during the Q cycle. Another important aspect about cytochrome C is that it is soluble in the intermembrane space. Finally, this leads me to mention that the illustration and animation that I will show of the Q cycle will be flawed because all the prosthetic groups and Q sites are located in the transmembrane region of the protein. This region corresponds to what's boxed in red on the screen. Since I want to make the dimensions of complex 3 be in line with what we've seen previously with complex 1 and 2, we'll have to consider this illustration instead of the more realistic view of the protein. Alright, now that we have all the necessary background knowledge to cover the Q cycle, let's consider the CoQH2 at the QO site. In the first phase of this cycle, CoQH2 gets oxidized to CoQ and releases its 2 protons and 2 electrons. The protons get expelled in the intermembrane space, whereas the electrons will part ways. One will go towards the intermembrane space, where it will serve to reduce cytochrome C, and the other will go towards the matrix to reduce CoQ and produce CoQH. After the first phase, we have now 1 CoQH molecule partially reduced at the QI site and 1 CoQ molecule fully oxidized at the QO site. We also have 2 protons pumped in the membrane and 1 cytochrome C with an electron, which can freely swap its place with the empty cytochrome C. In the second phase of this cycle, the CoQ that was just produced in the QO site gets released back to the membrane and a new CoQH2 replaces it. Here again, the electrons and protons get released, which produces a new CoQ molecule. The 2 protons go to the intermembrane space, 1 electron reduces a new cytochrome C molecule, and the other electron reduces the CoQH at the QI site to CoQH2. This newly made CoQH2 molecule can get released back to the membrane as well as the oxidized CoQ molecule in the QO site. In the end, the Q cycle produces one CoQH2 molecule from the QI site, two molecules of CoQ that were oxidized at the QO site, and two cytochrome C molecules that now each hold an electron. Additionally, the cycle pumped 4 protons in the intermembrane space. So, as you can see, the Q cycle is a pretty ingenious way for the electron transport chain to avoid wasting any resources, as it overcomes the limitation that coenzyme Q can carry 2 electrons, but cytochrome C can only carry 1. Also, it makes the most out of this process to boost the proton gradient, which will soon be important to consider when discussing oxidative phosphorylation. Finally, we reach the last complex, complex 4, which is also called cytochrome C oxidase. Based on what we covered during complex 1, 2, and now 3, what happens in this complex should be a little more straightforward to understand. For this complex, one of the main events that happens is the reduction of oxygen to water. For this to happen, we need electrons and protons. The protons will be supplied from the mitochondrial matrix, and the electrons will come from the cytochrome C molecules we have produced in the previous complex. As you may expect at this point, the electrons will be carried to oxygen by prosthetic groups, and in complex 4 there are two main types, cytochromes and copper atoms. Copper atoms work in a similar way to the iron atoms in cytochromes, as they hold a charge that can go from +2 to +1 depending on the presence of electrons. The mechanism by which oxygen is converted to water is pretty complicated, but the essence for us is that to make one water molecule, it requires that 2 cytochrome C molecules get oxidized and give away their electron. Throughout this process, for each molecule of water that is produced, 2 protons get pumped in the membrane. The mechanism of proton pumping is believed to occur through a channel, a little bit like complex 1, but the mechanics of how it happens are still unclear. You will notice that in addition to pumping protons, making water requires hydrogens, so this also contributes in creating a proton gradient. The final aspect that I want to mention about this complex is that you may see in different illustrations 4 protons reacting with 1 oxygen, making 2 water molecules. In this scenario, 4 cytochrome C molecules will be used and 4 protons will be pumped. What I previously shown is typically considered for figures because the Q cycle in complex 3 produces only 2 cytochrome C molecules, which fits better. In all, this concludes our detailed discussion on the electron transport chain. To quickly recap what we obtained in each complex, I want to make a diagram that illustrates the path of the electrons. Starting with complex 1, NADH gets oxidized to NAD+, and this serves to reduce one CoQ molecule to CoQH2. Additionally, complex 1 pumps 4 protons. In complex 2, FADH2 gets oxidized to FAD, and this also produces one molecule of CoQH2. No protons are pumped here. In complex 3, the 2 CoQH2 molecules get oxidized to CoQ, and in the process, one of their electrons reduces cytochrome C, and in the other serves to recycle one CoQH2 molecule. Each oxidized CoQH2 molecules pump 2 protons, and thus, the entire complex pumps 4 protons. In complex 4, the 2 cytochrome C molecules reduce oxygen to water and contribute to pumping 2 hydrogens in the intermembrane space. In total, the electron transport chain pumps 10 protons per water molecule made. One thing about this process that I haven't mentioned yet is that if we consider the formation of CoQH2 in complex 1 and 2, the reduction process also requires hydrogen ions in addition to electrons. As such, the protons that are released in the intermembrane space from complex 3 are basically the protons that complex 1 and 2 use to make CoQH2. Alright, back to this view of the entire process. Now that we know the function of the electron transport chain and how it operates to pump protons in the intermembrane space, let's take a look at the oxidative phosphorylation process in more detail. To do so, let's first consider the molecular architecture of ATP synthase, which is also known as complex 5. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.